a lot?
Yeah, because I've got a new lab program and we're gonna do a home run lab as well. All right. Now I'm going to get started. I don't know if it's taking Caleb so long. All right. So we're going to do the bell ringer for 211. We've got to finish collecting our data for the osmosis lab. And then we're going to do, I'm going to show you how to do Labster, but I may show you how to do that first. And then as you're working on that, I can have you come up and collect your data for that. There's cookies that'll be brought around and just completed on. So I'm probably going to jump to the Labster before we get to the osmosis, so I can get you started on that, and then one person at a time can come up and enter data for that lab. So the bell ringer first. All right, so passive transport. In passive transport, molecules move blank the concentration gradient.
They move up, not up, but down the concentration gradient.
So number one is down, so they're going from high to low, and that's like rolling downhill. In active transport, how do they move?
Low to high.
Low to high, so that's like going uphill. So think about riding a bicycle. If you ride it downhill, you don't really have to exert any energy. That's passive. You're riding uphill, you got to exert some energy to get that bicycle up. All right, 3, why do some molecules need extra help?
Some molecules need extra help, because like you just can't go to like, they need extra energy because you just can't go to low to high.
Well, this is still diffusion.
Mm-hmm.
So they're still going to go high to low.
Okay.
But why is it that they might need a protein channel to get through?
So they can break down the larger molecules?
No, think about size. They can be too
Big.
big. All right, they're large, or they are hydrophilic and can't go through the hydrophobic part. Number 4, what macromolecule helps in facilitated diffusion
Protein.
by acting as a tunnel, and that is protein. So number 4 is protein. Osmosis is the movement of water down concentration gradient. And you'll submit that.
Is that philic or common?
Philic, if I spelled it right,
Yeah.
hydrophilic, the water loving. All right, for those that just signed on, the answers, I've got it on the board, so you may not be able to see it. Number 1 is down. Number 2 is up. Number 3 is large molecules, or they're hydrophilic. 4 is protein. And 5 is water moves down the concentration gradient. So you're going to submit that. Let me get back in here. Now, before we get back to our osmosis lab, I want us to look at All right, I want you to go to the one that says Labster cell membrane. Go to Labster cell membrane. All right, so the directions here is that you will click on the link below to go to the online lab simulation. You will need to create a username and password this first time. Use the first part of your email and your email password. So when you click here, mine may automatically go in because it may be connected to my teacher.
Of the Stanford schools email?
Yeah.
Okay, so A and B, DRO 981, that's all no.
You're gonna have to click on, I believe, register
Yeah, I did that. Create account.
and create an account. That way I can at least, when it pops up on the teacher side, I can figure out who you are if you do that.
It's saying my password is too common, which it is for my Chromebook. It's 1, 2, 3, 4, 5, 6, 7, 8, but
yeah,
I'm
that,
gonna just put another password.
yeah, as long as you remember it. Most of ours here has a name with a number. No, I said, use that. I'm going in differently because I have created in a while. I thought I was going differently. Okay, it's working. All right, so
Your account has been successfully created. We have some questions to do on the rest of these files and you can activate your account.
the what?
They sent something to our email address.
All right, so click in your email address. And you may have to click on that. And then eventually you may have something here where it didn't recognize me as the teacher, so I'm going to just put my name in. So if you get to this point, once you click on what's in your email, make sure you do all this and save.
I ain't got nothing in my email.
This is the first time it's going to be this long. Can you pop up an email?
I'm still in here.
It should say activate Labster account. Now, you see all these nice little notifications here? You know how to turn them off so they don't log up your email account. When you get back in Canvas, and here on each home page of your course, somewhere in here. Let me realize this. There's supposed to. What is it there? Right here. You want your notifications. And if you turn it off right here for each of your classes, you won't get those in. And it'll clear out your. All right, where would jump gonna go here? Okay, somewhere down here. Set your reading. Where did it say it was sending your account? Okay, you did your school email, right? Oh, you need student in there, student dot Still doing schools all day. So, we need to, try to get these out. Here.
Mine is loaded at 7%.
Now, this will take a little while to load, but you should get a, now it may tell you, you may have to use a different, we type the password in, see if you can get there.
See, it just says like comes to this.
In front of the, right after the app. Student doc. Now notice how slow this is loading. Mine's gonna load slow because I have the video on. So if you close out other tabs, this is going to load a lot quicker. So I may have to stop presenting here and zoom that in. If I close out this, all right, and just because I closed it, it may still be presenting, I don't know. And let me click on this. Did it send anything this time? All right, so even though, guys, it's not going to be very clear, but you can at least kind of get an idea. It's still taking, it's on close out that too. Anything else? All right, so notice now that I've closed out of all the tabs but that one, it is loading a little faster. So know that your tabs have got to be closed out. It's got to be the only tab up there.
Even this one.
Okay, you may have to close out of all the other tabs. So mine is still loading. But what's gonna happen is we're gonna go inside this, it's set up kind of like, a 3D lab. And we're gonna be doing something with cell membrane and transport. So while that is taking place, we're gonna move to our other lab while that's loading. So I'm gonna take, let's see. We'll take Alex's, since Alex is online. And I'm gonna go through and I'm going to measure, like we did yesterday, our gummy bears, because we had put them in salt water after they had shrunk up. Oh, and my spoons.
Don't do that one with wet knees.
Yeah. Caleb, do you mind going to ask Miss Sawyer if I can have some spoons? Because I think I threw them away yesterday. I don't need. Huh? Yeah, but it's. All right. So I'm gonna take care of Alex's first while our lobster is loading. So remember, we are having to make sure that this little cup is, to find out what it weighs. Yesterday, it was about 2.4. See what's going on today.
You want me to go grab some extra?
No, 1 spoon's fine, because we're done after this. We don't have to do anything else with it. All right, so today the cup is showing 2.2, so I'm gonna write this on the board. So this is for Alex. Amber, do you mind getting Alex's paper out so we can get his information? You wanna get, Josh's right there, and we'll write his on it when we do this. So I'm gonna do the tap water first. And I don't know if you can see it here, but we have just a teeny little gummy bear again. Yesterday it was real big. And so we're going to, all right, 10 is too much, so I know it's going to be over this way. So in tap water, it mixed with salt water, the gummy bear shrunk back down to, no, not quite.
Not all the way, but. Well,
6.3. So I'm going to subtract. Use some more. All right, so 6.3 minus 2.2, so 4.1 grams. So Amber, under tap water, if you'll write 4.1 grams for Alex. All right, I'm going to,
Under salt water, you mean?
oh yeah, but it's under the tap water side, yeah. Red, yeah. All right, so now we're gonna do the green. Now remember yesterday we had a little problem with his green one. I kind of mutilated it. So I'm going to need to brush back up and make sure my cup is the same weight. All right, 2.2. So here's part of that one. I can't get the other part out. It's too much in the water. All right, so we're going to weigh this. This time I got well, 5.7, but remember I didn't have all the gummy bear. So I'll need to remember that to make a note of that. 2.2, so that is 3.5 grams for the other, or if you'll write that down. All right, which one of you want to do yours next? And then we can just pour these out and we're done with this. So they did shrink back some with the salt. And I'm at 72% on those other things here. Well, let's get a. Oh,
And you got safes back here.
huh?
You got whole safes back here,
No, they're chemical things, so.
basically.
All right, is that 2.2? You'll have that on 2.2 there? Yep. Okay.
Too much, right?
Yeah. Caleb, I'll have you do one of Josh's and Amber can do one of Josh's.
Which ones are getting these from time?
Yes, because yours is on the shelf, right?
7.7 grams.
All right, 7.0, 2.2, all right, so that is 8. Okay, 4.8 grams, which one is that for? Is that tap water?
Distilled water.
Huh?
Distilled water.
Distilled water? All right. And then you'll need to do your, other one. What's there?
Two, yeah.
Oh, no, just a little bit over.
Yes.
This one. It's gonna be 2.3.
Mm-hmm.
Oh. Good. Good. 2.1.
Yeah.
All right, so this one will subtract 2.1.
Eight.
All right, so 8, that's gonna be 9. 5.9 for your tap salt water. 5.9, yeah. And then if, this is Josh's, if you'll do one of Josh's, I'll leave yours up here. And I'm just gonna use one of these as long as I'm doing it. Might be a little bit more because I'm using a cup. As long as we. Okay, what is it? 2.2, okay? And you're
Did
doing
my
Josh-
did my gummy bear come back together?
Well, it's
Some type.
it's smaller.
Oh snap!
She's getting ready to weigh it for you, so we're gonna write it down. All right, so back here, my screen is finally on about 99%. So by the time we get this weighed, it may be loaded. All right, so the red gummy. Josh. That is.
Here's some that you don't want. Don't dissolve me. Miss Noel, help me.
Let's see.
Where is it?
It dissolved. All right, we're just gonna pour it in like that. Josh, yours kind of dissolved. All right, let's.
Wow.
Go back to the stuff.
And Josh, we're getting cookies today for working.
Bro, yes, you're not even here.
I'll pull up.
Yep, 7.5, so 7.5 minus 2.2 is 5.3. So his red one was what, distilled?
Yep.
All right, Caleb, if you'll take care of his green one there. And, oh, who's that?
Y'all, look at my makeup. Okay, so this is how I did it. No, little red eyeliner.
Amber, get your ass.
Oh, got it nice.
You said what?
Huh?
Good up close of you.
Yes, a good up close.
All right. So write down your numbers.
What kind of cookies y'all getting?
Miss Sawyer made chocolate chip.
Good cookies. And they're
Oh.
big.
Goodness.
Yeah, you're
Should have come today.
not even here.
Hey, look, I woke up and I really wasn't feeling good. You know, my nose was running a little more. It was kind of stopped up.
Do you think we care about your kids?
Yeah,
We don't.
y'all do.
Maybe she'll cook something else next week.
Yeah, hopefully I'll beg her. I'm be like, cook some cinnamon rolls.
All right, now see how much that one is. Your green one stayed together a little better.
I think my gummy bears stayed together the best. They had their shit together.
Okay, 7.1. So the other one is 7.5. All right, so 4.9 for that one, so tap water. So we'll get that written for Josh. All right, Caleb, now you do yours. Pour that back in his cup. Oh, yeah. Up here. Where did we put Josh's paper? Over there. You want to write these down? For, so the green is this one.
4.1.
Uh-huh. What is, it was a 2.2?
Red is 500.
All right. It's that for Caleb. What else I can do? Get this one here. Oh, jeez.
Oh my gosh, I'm trying to make my parents come out of this person's head and it's not working.
Well, that one stayed together very well.
So do my green one, look at it.
Yeah.
8.
Right. All right, 5.8. All right, now your next one. Guys online, remember I need makeup work tomorrow.
Oh, my grace.
Since you guys have some paper copies, if you can't get it to me tomorrow, you got to
Can
have
I-
it.
Can I drop it off in the
Okay.
morning?
Yeah,
Okay.
if you can't get it dropped by tomorrow, you need to, you can bring it Monday, but that's the latest.
All right.
I would prefer it tomorrow.
Pull yourself up.
Is that Alex? Did you say something, Alex?
I think they're texting.
That's the pictures, Keith.
6.
6.5.
These two gonna very stay together pretty good.
All right.
That's good.
So get your data down, and then we can. Well, my whole point was for us to do a lab, but if it's not gonna load, we've got a problem. If y'all's load at all,
I got 1%.
yeah, see, mine's stuck at the 1% too.
Oh, man,
And I
don't.
don't know why. Oh, mine did it, okay. I can at least show, hopefully yours will pop up, as well. I did refresh and I finally went. So, looking at this, we're going to, you click start. I gotta have volume. How do I get my volume on here? So I don't know if that picks up at all online. But this is a simulation, and you'll always get this to start with. And you really don't have to do anything up here because you have speakers that are set up on there. You'll click continue.
What was my data?
Right down here, tab and distill. So, we're gonna walk through this first time. We're gonna do A. So if this is the first time, on you would have to use your mouse. This is touch screen. So, this is your lab pad. It's gonna have information under theory. It may have pictures under media, and it kind of gives you some explanation under mission. So you can lower this by clicking anywhere in the background.
Well done. When your lab pad is lowered, you can listen to me without having to click continue. You can interact with highlighted objects. Arrows help you in finding their location. Now click on the corner of your lab pad to raise it.
And so there's an arrow pointing, and you're going to click on it. So click continue. You can access all relevant information from the theory. So you can click on the theory, and it should give you information here. Again, loading if you don't have a good connection, as it seems to be right now. It's not going to load, but it has written information, background information about the concept. To scroll down, you would use your mouse or wheel to drag the gray bar on the right side, just like you would on anything right here. right in here. To scroll down. Well, I'm trying to click down there. So unfortunately, you've got to have good internet for this, it appears. I did it the other day and it was working just fine. Click here to confirm you know how to scroll down. You can rotate the camera by clicking and dragging your cursor to the side. So the camera was here, where that camera went. All right, so I think it's Dr. 1 is the name of the camera. Your score will be up here, and this will register on the teacher side, and that's what I'll record for your grade up here. And it kind of tells you the progress as you go through here. Here's a calculator if you need it. The media tab will give you images, and I can click here and we can try. So here's some images here to give you an idea, but there'll be more dealing with whatever the lab is about. The mission, you can track your process. You can go there to see an overview of your work or load from a checkpoint. You can see your progress. So we're just going to say yes. So here, did y'all ever load?
Nope.
Okay, so notice it says here, find the lab code and click on it. So it's pointing this way, so I'm going to have to drag around this way.
Click on the lab pad for more information.
And so I've got my lab coat,
Find a glove box and click on it to put on the gloves.
and so it's going to say glove box.
You can explore the lab by hovering over items in the lab to find out more about the science that happens here. Dr. Hacker will be with you momentarily.
And so then, so like
Hi there. Welcome to our synthetic biology lab. I am Dr. Bio Hacker, and I see you have already met my AI assistant, Dr. 1. We are trying to create synthetic cells that make useful compounds powered by photosynthesis. We already have a prototype of our synthetic cells, but we have some work to do to improve the health of the cells and the efficiency of the process. Just like real-life cells, our artificial cells contain a nucleus that holds the DNA, and this DNA is transcribed to RNA and then translated into proteins. The cells can even send signals to other cells by releasing proteins into the extracellular environment.
all right, so it's wanting me to go this way. So here's the holographic table.
Activate the hollow table.
So they want us to activate it. All right, so here is the cell membrane. So this is the bilayer. So as you hover over it, you can see the different parts, and it tells you what it is. So it is a lipid bilayer that has hydrophilic water-loving heads. These are the circles. And you have hydrophobic tails that are pointed toward each other there. So you'll go next and hover up here. Well, thought it would have more. The most abundant lipids are phospholipids, but the cell membrane also contains cholesterol, glycolipids, and other lipids represented in different colored spheres. So these different color spheres represent different color lipids.
The cell membrane forms a compartment or cell that is separate from the extracellular environment. What is the other main function of the cell membrane?
All right, so look up here since yours isn't going. What are some things here? Is it for facilitating migration of the cell through body? Is it for providing a rigid structure that gives the cell shape? Is it for compartmentalizing organelles within the cell or controlling the transport or substance inside and out of the cell? Which one? And if you don't know, we can view theory to see.
What is the cell membrane?
If don't do that, we're having to do this together. B, so,
Great job. The cell membrane forms a selective barrier between the inside and the outside of the cell as it controls which molecules can move in and out of the cell.
all right, so notice that we got 10 points toward our 100 here. So then we're gonna continue.
Due to the composition of the cell membrane, some molecules can cross the membrane more easily than others. This is called relative permeability. Which type of molecule is least able to cross the membrane without transporters? There is a poster in this room that will help you, or you can click View Image in the lab pad.
All right, so look at the question again. It says least able. All right, so we have ions, small charged, uncharged polar molecules, large uncharged polar molecules, or hydrophobic molecules. So let's click on the image and so low to high, we have ions, large uncharged, small hydrophobic. So least likely, I would assume it would be the one over here with low membrane permeability. So it looks like it might be the ions. So let's see if I've read that chart right. All right, so you kept your ion, the prize. All right, ion charge makes it hydrophilic, and so it's going to have a hard time going through. All right. So notice the arrow here says, let's head over to the bioreactor workbench and grab a sample tube from the rack when you get there. The arrow is pointing this way, so I'm going to click and drag. Keep going. There it is, bioreactor, so I'll get fine. Oh, here we go. Oh, there's the tube. So they poured the tube in there. Or got a sample rather. Place it back here. All right, select a molecule using the buttons on the right to shoot it at the cell to determine whether it can cross the membrane. All right, so we're going to choose, let's try bulk. All right, so was it able to cross it? All right, let's do charge. That would be like an ion. No. All right, notice that one went through. All right, as you can see, an aquaporin has now appeared in the cell membrane. Which type of molecules are aquaporins? Aqua means water. All right, so that means we would choose water here. So here's an aquaporin. That means we can select the water. And the water can go through if you have this.
Select a molecule using the buttons on the right to shoot it at the cell and determine whether it can cross the cell membrane.
All right, so let's do polarity. Well, notice it went through the channel protein.
Good choice. You unlocked a carrier protein. These proteins bind molecules on one side of the membrane and undergo a conformational change to release it on the other side. Select the type of molecule that might enter through a carrier protein to launch it at the cell. Which type of molecule would use a carrier protein to enter a cell?
All right, these channel and carrier proteins mainly transport molecules passively. Active transport, on the other hand, needs energy. Why does active transport require energy? Which one am I going to choose, guys?
Active transport requires energy to transfer molecules. Like, wait, no.
We'll try this one.
I think so.
Move your.
Oh.
No, it was right.
Mhm.
We didn't learn it as electrochemical, we just learned it as.
Now, that's the thing, I had to
Gradient.
freaking, I
So the
just
key
see.
was gradient here.
You have to see.
You are building transporters like a protein. You managed to find out that only small hydrophobic molecules can enter the cell without specialized transporters. Water enters the cell through aquaporins, and bulky, polar or charged molecules need a channel or carrier protein transporter. Now let's look at some subtypes of these transporters and how they are controlled. You now have more types of transporters on the cell. Your goal is to choose the correct transporter for the type of molecule we are going to launch at the cell.
All right, so here are different transporters. So notice I clicked on Fury. And we've talked about the aqua, the channel. Notice channel proteins will do voltage gated. Ligand gated means it needs another molecule to trigger it to open, or mechanically gated. You've got carrier proteins. You've got active transport that needs energy. So if we need to come back to look here, we can. So now we're going to continue.
You are now launching an ion. Which 3 transporters can let ions into a cell? Click on them.
All right, what's going to let an ion in? They're charged. Voltage. All right, what else? We'll try it. Yep,
Anti-porter? No, aquaportin.
so that was the water.
Oh. What was the one guy?
Try this one. Oh, it went in, didn't it?
You've got this. Ion channels can be mechanically gated, voltage gated, and ligand gated. I think you are ready to have another type of molecule and a few more transporters and mechanisms thrown into the mix. Now, you are launching 3 molecules that require carrier proteins to get into a cell. Click on the correct 3 transporter proteins for these molecules.
Well, it's not a carrier. So a carrier, I think, may have to have the word porter on it. So if we roll over it, see it says carrier molecule. Let's click on that one. Let's see what this one does. All right, and I think it is this one.
Click on the lab pad for more information.
Now, how I knew that because
Go
I
you.
wasn't,
Carrier transports can be uniporters, which only transport one molecule in one direction, symporters, which transport multiple cargoes in the same direction, or antiporters, which transport multiple cargoes in opposite directions. Let's add in another type of cargo molecule.
all right, I knew that was because I knew the term port means to carry. And so if you weren't sure.
Now, you are launching molecules traveling against a chemical gradient. Which 3 transporters could get these molecules into the cell?
Against. All right, if it's going against, that means we need,
Anti-porter.
we need ATP, right? Against,
Yeah, ATP.
so probably ATP is going to use energy. Let's see, look at this one, I'm hovering over it, it says uses electrochemical gradient. So I can click on that. What is this?
Click on the lab pad for more information.
And the light driven.
Awesome work. Transporting molecules against the electrochemical gradient requires energy. These molecules are transported by ATP-driven pumps, light-driven pumps or coupled transport, which uses the transport of a molecule traveling down the electrochemical gradient to drag another molecule against the electrochemical gradient. Which of these options is not a mechanism to control the opening and closing of ion channels?
All right, so let's look at this. Which one is it?
He's still in that one?
He was online, he's online.
Okay.
They was still in that one?
All right, guys, we gotta answer this question. Which of these is not a mechanism to control the opening and closing of ion channels?
ATP. Oh, no. No.
Opening and closing.
Yes, ATP, that's the only one I'm familiar with.
Well, it involves energy, so that's gonna, it's not a mechanism to control. And we can try it. I thought all of these, because this means something binds to it and it'll open. This one might be this. I don't know, so we can go here and all right, gated channel through to membrane that opens and closes in response to a binding, mechanical, or voltage. So let me check ATP. So uses energy against pumps, molecules. So maybe, you may be right, let's try ATP.
Excellent. Ion channels can be voltage, ligand, or mechanically gated. ATP is one of the drivers of active transport, along with light and coupled transport. How does coupled transport allow a molecule to be transported against the electrochemical gradient?
How does coupled transport allow molecules to be transported against the electrical?
Would it be, use energy since it's going against?
Yeah.
Almost. Remember that coupled transport is also called co-transport. Check out the theory tab in your lab pad if you don't know the answer.
Co-transferring molecules travel down.
That's right. Another molecule going down the electrochemical gradient drags the molecule of interest at the electrochemical gradient. This other molecule can either be traveling in the same direction in a symporter transporter or in the opposite direction in an antiporter transporter. You don't just build transporters, you know how to operate them too. Through these gating mechanisms, the cell controls which molecules go in and out of the cell. Let's go back to the lab to see if Dr. Hacker was able to use this information. Teleporting back to the lab. Wow, we sure learned a lot about transport proteins from the virtual cell. Dr. Hacker has used your learning from the virtual cell to insert transporter proteins into the membrane of some synthetic cells. Let's test if these proteins actually transport molecules across the membrane using fluorescence microscopy. Enter the lab.
All right, so we have to enter the lab. That means I think we have to go in here.
I love shade. And I'm not done my learner. I'm gonna make it.
Great, let's start at the biosafety cabinet. For the next mission, you need to use a pipette to transfer substances. Would you like to play through a short tutorial to learn how to use the pipette? The pipette is used to transfer very precise volumes from one container to another. Pick up the pipette.
Yeah.
All right, guys. Okay, so this is an actual pipette that you would use in a real lab. It's not like the plastic ones that we have. So you would get it.
Click on the box with the pipette tips to
and
pick
it has
one
to
up.
have a tip.
The disposable pipette tips are sterile plastic tubes. Make sure you only use a pipette tip once, otherwise you might contaminate your samples. Click on the water bottle to fill your pipette tip with water.
Okay, so notice this says contains H2O, which is water.
In the real lab, you would need to adjust the volume on your pipette. This is a bit tedious, so I will automatically set the correct volume and you don't need to worry about it in the. Now transfer the water to the tube on the tube rack.
Alright, so we have to transfer it here.
You have successfully transferred the water. Now click on the trash bin to discard the used tip. Now you know how to use the pipette. There is a theory page about pipetting on your lab pad. Put back the pipette on its holder. Dr. Hacker has already prepared the cells. You can find them in the incubator at the microscope workbench. You can open the incubator using the open close button with the arrows. Great. If you pick up the 6-well plate and take it to the bios.
Here's the microscope we're going to be using.
Feels like one of the games I played,
Yeah.
the Walking Dead game.
Well, now I'm trying to figure out what I'm supposed to do next. Put the hologram on the down. Well, I did that. It's there.
Maybe click the little eyeball.
Click on the lab pad for more information.
What if you're not telling me anything? Click on the 6-well plate. You get up, and click the biosafety count. Put this up. Biosafety count. What's that? Here's the biosafety. You see an arrow telling me where I need to go?
No, that animatronic girl tripping.
I need to go to the biosafety panel. It's the biosafety panel.
What's that over there?
That's the.
How can I help you? Is it because you don't understand the theory or because you don't know how to continue with the simulation? Oh dear, I hope my simulation isn't glitchy. Have you checked the additional info that you can see when you lift your lab pad? If you are totally stuck, you can always reload the mission from the last checkpoint. Just click on the mission tab and click on the load checkpoint button.
Usually it gives me an arrow. Ah, there we go.
We will be using the fluorescence microscope. Can you figure out which substances from the biosafety cabinet you need to add to the cells to measure transport across the cell membrane? If you use the pipette to add the substances, I'll make sure to automatically set it to the correct volume. Add fluorescently labeled cargo molecules and the membrane dye to the cells in a well using the pipette. Remember to use clean tips.
Oh well, I'm stuck here. Okay, so.
Click on the lab pad for more information.
ask and you shall receive. Congress again. You either have mom or start the same high school, so break out with them and days.
Your blood lines in case you think they're weird.
Did you elect for that today?
I'm fine. We got kind of stuck.
Yeah, that's fine.
Did you manage to figure it out? Use the pipette to add the green cargo molecules and the red fluorescent dye to the cell plate. If you are unsure if you have added a substance to the cell plate, hover over the tooltip to hear what substances it contains. Remember to add a sterile tip to the pipette for each substance. You must dispose of the used tip.
Well, yes, I'd like to put it in there, but you don't really put it in there. There we go. I guess you have to know which one you're supposed to put it in. There. All right, guys, if you're in line, you can sign off.
But look at my jumpers.
Great. You are a real lab genius. Great job. You have prepared the sample correctly. It now contains the cells with a transporter protein, cargo molecules labeled in green, and a red dye to label the cell membrane. I will go ahead and prepare the rest of the... Why do we need to label the membrane?
All right, well,
You are great at experimental design. Awesome. Now let's put the pipette back and take the cells to the incubator at the microscope workbench so the transporters can do their job. Open the incubator and place the 6-well plate inside. The cells need to incubate for an hour. Luckily, we are in a virtual lab and can speed up. Wait for the incubation to finish.
We know that
we had some.
Let's take the cells to the fluorescence microscope.
You need a break or anything?
Cookie, sir?
Not right now.
Cookie, ma'am?
Don't forget your.
You're welcome.
The fluorescence microscope uses lasers to excite different fluorophores. And are the molecules able to cross the membrane?
Have another sandwich? Yeah, you want it?
Yeah, whatever. We ain't got.
I won't eat it. I always order, but I don't eat it. You want this meal food? Oh,
Put my sandwich in the microwave and you put yours in there.
here. Would you like extra milk?
No, thank you.
That's right. The green fluorescent. Dr. Hacker used a type of transporter called an ABC transporter from bacteria. Eukaryotes like humans also have ABC transporters. Since we are using synthetic cells here, we could use transporters from either system. Interestingly, while both prokaryotes and eukaryotes have ABC transporters, they are not exactly the same. The principle of membrane transport is the same across all.
Like, it just exited.
All right, let's go over to the wall screen now.
So I want y'all to try this at home and see if you
It'll
can
probably
it'll
work
close.
because the Wi-Fi.
try to work. Let's take our, let's figure out the wall screen.
Be right back.
Oh, this?
Transport across the membrane can be highly specialized in differentiated cells. This allows the cell to execute its role and work together with other cells in the tissue or organ system it belongs to. On the wall screen, you will find transport proteins that are highly expressed in certain human cells or organs in their role. Let's see if you can match